Hello and welcome to this Infection Basics video. I'm Dr. Aled Roberts, and in this video we're going to look at microbial pathogenesis. Specifically, we're going to look at the infection process and how microorganisms invade and colonize a human host. And so the process of pathogenesis starts with exposure to a potential pathogen. Essentially, the pathogen has to get into the body of the host somehow. And there are several common routes they can use. One major route is the respiratory tract, where pathogens are inhaled as aerosols or droplets through coughing, sneezing, or even normal breathing. Another important route is the gastrointestinal tract. Here, pathogens enter through the ingestion of things like contaminated food or drink, and if something survives the stomach acid and reaches the intestines, it can establish infection there. The urogenital tract is another potential entry point, often through sexual contact. The skin usually acts as a strong physical barrier, but it can be breached, and so small nicks, cuts, abrasions, puncture wounds, surgical incisions, or bites can provide an entry point for microorganisms. Then closely related to this are the mucous membranes, such as those in the eyes, nose, and mouth. These surfaces are moist and exposed, making them common entry sites if pathogens come into contact with them. Now, it's not listed here, but there are also some less common routes, such as transplacental transmission, where pathogens move from mother to fetus through the placenta. Now, all of these routes are collectively known as portals of entry, which is a term that simply describes how and where a pathogen enters the body. So let's look at some examples. Take the alpha influenza virus influenzae, for example. It does not cause skin infections. The skin simply isn't a suitable environment. It lacks the right target cells, the keratinized outer layer is difficult to penetrate, and the necessary receptors aren't present. However, this virus is well adapted to the respiratory tract, where those conditions are met. A similar principle applies to Streptococcus pneumoniae. This bacterium does not cause gastrointestinal infections because it's highly susceptible to stomach acid and is killed before it can establish infection in the gastrointestinal tract. In contrast, it is very effective at causing respiratory tract infections where it encounters a much more permissive environment. Finally, Chlamydia trachomatis is another good example. It cannot infect intact skin for the same reasons: no appropriate target sites and the effective physical barrier of the skin. However, it's specifically adapted to infect the urogenital tissue, leading to sexually transmitted infections. And so the key takeaway here is that exposure does not guarantee infection. For infection to occur, the pathogen must enter via the right route and encounter the right conditions to survive, attach, and replicate. Now, following on from entry and exposure, the next important consideration is infective dose. This refers to the minimum number of viable pathogens required to cause infection within a susceptible host. Now, in infection biology, this is often described using the ID50, which is the dose at which 50% of exposed hosts become infected, and it gives us a useful way to compare how infectious different pathogens are. Now, it is important to stress that infective dose is not a fixed value. It isn't just a property of the pathogen alone, but of the interaction between the pathogen and the host. And so on the pathogen side, infective dose is influenced by virulence traits such as toxin production, capsules, and adhesins. Then on the host side, we have the microbiome, the immune status, and whether they have been previously exposed to the pathogen. And so together, these factors explain why the same exposure can lead to infection in one person, but not another. Now, infective dose varies wildly between pathogens, from very low to extremely high. Some pathogens have a low infective dose, sometimes fewer than 100 cells. So as an example, Shigella species and Escherichia coli 0157H7 are both acid resistant, meaning only a small number are required to cause infection as they resist the effects of the host stomach acid. Others have a medium infective dose, typically in the thousands. So as an example, Salmonella enterica and Campylobacter jejuni are partially acid sensitive, meaning larger numbers are needed to survive the trip through the stomach. Then at the other end of the spectrum are pathogens with a high infective dose, often over 1 million cells. And so things like Vibrio cholerae are acid sensitive and usually require ingestion of heavily contaminated water, while Staphylococcus aureus is kept in check by effective host barriers, requiring lots of cells to overpower these defense mechanisms. Now importantly, infective dose is context dependent. Vibrio cholerae requires a much lower dose when stomach acid is reduced, such as if the host was using antacids, whereas some E. coli strains that lack the key virulence factors, such as O157H7, tend to require a much higher dose. Okay, so once an effective dose is reached via the correct portal of entry, it's important to remember that the infection has not yet started and disease has not occurred. At this point, the pathogen must first associate with the host cells, and that initial contact happens through adhesion, which is the first essential step in establishing infection. So why is adhesion necessary? Well, it's because the host is constantly trying to remove potential pathogens. It doesn't want to be infected, and so it continually uses a range of physical clearance mechanisms to stop infection. Cilia and mucus sweep microorganisms out of the lungs, tears and saliva wash them away from the eyes and the mouth, urine flow clears the urinary tract, and peristalsis moves microbes along the gut, each of which are first-line innate defense mechanisms. Now, it's never that easy. To resist being removed, microorganisms rely on what we call adhesins. This is a general term for molecules that promote attachment to the host cells. Now, adhesion is therefore a direct countermeasure to these innate defenses and a critical early step in infection. Now, adhesins come in many different forms depending on the type of pathogen. In prokaryotes, such as bacteria, adhesion typically relies on fimbriae, pili, flagella, and capsules. Eukaryotic microorganisms, including protozoa, use different structures such as cilia, suckers, hooks, and barbs. Whereas the viruses, they rely on surface spikes made of proteins. Now, despite the differences, the principles are the same. Adhesion enables pathogens to remain in place long enough for the infection process to begin. And so if we look at some specific examples, fimbriae are widely used by bacteria. Pseudomonas aeruginosa, for example, uses them to adhere to the respiratory mucosa, while Salmonella, Vibrio, Escherichia and Shigella species, they use them to attach to the intestinal epithelial cells. Bacillus anthracis has a particularly sticky capsule that allows adherence to a range of host cell types. Suckers are used by various Giardia species to attach firmly to the intestinal surface during giardiasis infections. Hooks are seen in Treponema species, where the tapered ends kind of embed into host tissues during the syphilis infection. We have viruses which are dependent on surface spikes, so the influenza virus binds sialic acid receptors on the respiratory mucosa, while HIV will typically attach to specific receptors on white blood cells. Now, adherence is so critical to infection that most pathogens rely on multiple adhesion mechanisms rather than just one. Now, following on from adhesion, the next stage is colonization and invasion. This is where we get the establishment and maintenance of a pathogen at a specific location, allowing it to persist within a host. Now, in some cases, this can be followed by invasion, where the pathogen penetrates tissues or spreads beyond the initial site of infection. Now, at the same time, the pathogen must survive ongoing host defense countermeasures, making successful colonization a critical step in disease development. And so one of the primary host defense mechanisms is phagocytosis by leukocytes. These cells are continually monitoring host tissues actively searching for invading microorganisms and removing them once identified. Now, pathogens are not passive in this process. They effectively anticipate this response by the host and possess a range of defensive strategies that help them avoid detection, resist killing, or survive once encountered. And so many pathogens produce anti-phagocytic factors that directly interfere with the phagocytosis process. For example, protein A in Staphylococcus aureus prevents effective opsonization, while the N protein in Streptococcus pyogenes blocks complement deposition, reducing recognition by phagocytes. Some bacteria go further and produce leukocidins, which are toxins that actively kill cells capable of phagocytosis. So Staphylococcus aureus produces LuC-A-B, which targets neutrophils and macrophages, while Streptococcus pyogenes releases streptolysins O and S, which have a broader leukocyte-killing effect. Another common strategy is the production of a capsule or slime layer found in many bacterial species, and this creates a slippery physical barrier that makes phagocytosis mechanically difficult. And finally, some other pathogens use an intracellular survival strategy, and so phagocytosis is actively encouraged. And so organisms such as Legionella species and Mycobacterium species, they are able to survive and replicate within white blood cells, allowing them to persist and be transported around the body largely undetected. And so once a pathogen has successfully entered the body, adhered to host tissues, established residence, and evaded immune clearance, the conditions are finally in place for growth. At this point, the pathogen is no longer simply present, it is established within the host. And so for bacteria, this usually means replication through binary fission, allowing the numbers to increase at the site of infection or within host cells. For viruses, growth is through the burst concept, which involves entry into host cells, replication of viral components using the host cellular machinery, and release of new particles, often through cell lysis. Now, regardless of the mechanism, the goal here is to rapidly increase the number of pathogens within the host, completing the process of infection within the host. And so, as a quick recap, first, the host is exposed to a pathogen, but exposure alone is not enough to cause infection. The pathogen must then successfully adhere to host tissues, allowing it to remain in place rather than being removed by physical and immune defenses. Once attached, the pathogen can then colonize the host, establishing itself at a specific site, and in some cases, invading surrounding tissues. This step is critical as it allows the pathogen to persist despite ongoing host countermeasures. With colonization established, the pathogen can then begin to grow and replicate, increasing its numbers within the host, and at this point, the infection is fully set up. Virulence factors are activated and disease can begin. But that is for another video. Which brings us nicely to the end of this one. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.